This brings me also to nutrition, because when we're talking about establishment of memory and the ability to remember something, what you saw was that we can have these physical changes in the brain when we establish memory, and we're going to talk more about it on Thursday. So basically, and I mentioned it a little bit a while ago, when we have the contacts between two nerve cells here, so a synapse being created, so you have one nerve cell over here, you have the other nerve cell here, you have the cell membrane here. You know that it's this bilipid layer built of mainly phospholipids. Now, if you look at the ingredients which go into creating a membrane like this, if you want to establish memory in the sense that you establish a new connection, a spine which is growing out and creating a new synaptic connection here, you need basically uridine, you need choline, and you need DHA. DHA is docosahexaene acid, which is an omega-3 fatty acid. And in the brain, it's about 60% of the cell membranes, which comes from this compound alone. So that's interesting in itself. But now, if you look at a rat, which is just eating normal rat food, what you see is that the spine density, the number of synapses in a certain area of the brain, is about here a little bit over 60 for each 50 microns. Now, if you supplement the food with a little bit of uridine, a little bit of DHA, and uridine and DHA together, you see increasing number of spines, which means that rats don't eat enough uridine and DHA in order to create as many spines as it could have if it had had these compounds in its food. So simply by supplementing it, you can boost the number of spines being created in these rats. So what does that look like in humans is a question. First of all, what is DHA? So as I said, it's docosahexaene acid. It's an omega-3 fatty acid. It's being produced by these guys. Latin name Schizocrucium. What it is actually is that it's small monocellular organisms living in the oceans, rødalger, red algae, which are being eaten by fish. So these are the ones which produce DHA, and of course very small amounts in each of them. They are being eaten by small fish are being eaten by large fish, and then eventually big fish like salmon, tuna, mackerel, similar fish end up having quite a lot of DHA in their muscles, which is part of the reason why these fish are able to swim in very cold waters, because DHA and some other fatty acids ensure that their muscles are nice and soft, even when it's really, really cold in the ocean. That's also what makes them, to a large extent, taste good, and also makes them have the color that they have. It all comes from these red algae eventually. So what is important for us is that we actually have to eat fish and similar compounds in order to get DHA. So our body is not equipped to produce any significant amounts of DHA. We can produce a little bit based on other omega-3 acids, but it's relatively limited. So we need to eat it in order to get it into our brain. The main source that we have for DHA is fish. It's hardly found in any other substances, which means that if we don't eat DHA, we won't get the substrates for the nerve cells. There's a little bit, and I don't know, what is it called? Raps? Granola? Really? Okay, I didn't know that. So there's a little bit of DHA in granola, but not very large amounts, and you have to eat quite a lot in order to get any significant amount. So choline is also essential. Our liver will degenerate if we don't eat it. But I'm not really worried about that because there are lots of sources for it. So liver, meat of many kinds, lots of vegetables, fish, milk, whatever, many different sources for it. Uridine, I'm also not really so concerned. There are lots of sources, raw sugar, tomatoes, yeast in beer, whatever, broccoli, lots of vegetables. So we're probably getting enough uridine and choline. The worry is the DHA, because we almost only get it from fish. The amount of fish that we need to eat in order to get enough DHA to cover the need of the brain corresponds to actually eating fish at least for 1 meal per day, and from 1 major meal, that means dinner, 2 times per week. So the equivalent in DHA is about 200 to 500 milligrams per day. And it's relatively few of the fish oil products which contain that amount. So if you want to buy fish oil products, you have to look out for it. So what happens to rats if you give them DHA and uridine? So again, the Morris water maze, you have control rats here, which are not eating uridine, DHA. They start out a little bit more than 60 seconds to find the platform. After 16 trials in total, they find it within 30 seconds. Those who get uridine and DHA start out by a little bit under 70 seconds, and they end below 10 seconds. The only thing that has happened here is that they have had a supplement of uridine and DHA. That's all it takes. And this increased ability to find their way in the Morris water maze is quite well correlated to the number of new spines that they make per 50 microns. So just giving a little bit of DHA is all it takes. In this case, we also know what the mechanisms are. So it turns out that DHA actually stimulates very much the same pathways that exercise. So again, BDNF and other factors like insulin growth-like factor 1 are involved in signaling the changes that take place in these spines. So the spine formation, which is simply a genetic program which creates the membrane, creates the receptors to create a new receptors, are all signaled by BDNF and IGF-1 basically. So when looking at the combination of DHA and exercise, as you can see, control rats, rats giving DHA and rats doing both exercise and getting DHA works much better than any of them alone. So the right nutrition together with exercise actually does wonder, at least in rodents. The question which is still hanging here is, does this also work in humans? So exercise, I would say yes at the present time. In terms of nutrition, we don't really know. It's quite clear that in newborns and young children, there's quite a lot to be gained by making sure that they get the right nutrition when the brain is still developing and creating a lot of new connections. It's not as clear for elderly subjects and maybe patients with brain damage, but there's quite a lot of research which is being done. We have also been doing quite a lot in relation to children with brain damage, and we had a study last year. One of the master students did a study on children from one of the schools here in the Copenhagen area, where we gave substitutes of DHA, uridine, choline for 2 times 6 weeks, total 12 weeks, and actually did see significant improvements in cognition. It was made as a double-blind placebo-controlled study, so we had no idea who got the active substance and who didn't. The children didn't know when they got the active substance and when they didn't. So it's a highly controlled study, and still we ended up with actually a reasonably good effect. Not as big as we had hoped for, but at least an effect. So it seems to work amazingly.